Hello everyone, I'm Meena Choi, an Associate Research Scientist at Northeastern University. Today I will present MassIVE.quant, which is the community resource of curated quantitative mass-spec-based proteomics dataset. MassIVE.quant has been a recent development that we've integrated into MassIVE that is this global resource designed for systematic sharing of the quantitative mass spec data. We've run quantitative mass spec experiment and analyzed the data set. We know there's a lot of the step to take care of. There are many ways to search in peptide and then protein identification. We can use the different searching tools and we can use the different quantification tools. We can have a different mass spec experiment. All of these things require different configuration on how you run the software and how to use it. So it is very helpful to have a place where you can deposit all analysis in a manner such that you can go back and reproduce this identification searching and then quantification and statistical reanalysis. Because this is the first step and the very important step for reproducible research. MassIVE.quant provides a new infrastructure for sharing this kind of information, such as new collections of the quantitative information, and then branch structure for reanalysis, and compilation of the fully public data set, and then as a promising perspective with this new aspect, the MassIVE.quant to provide executable the workflow, and additionally, that all the documentation and video tutorial for each workflow and step and case studies are also available. Previously, the most repository resources have focused on the spectral identifications. For example, there are collection of the raw files, and then the database, and then identification result. If identification result is submitted with the raw file, it is a complete submission of dataset. Other files for next analysis step were not required and classified to others. Therefore, it is very hard to find or search the data set including quantifications and the metadata. Now, the MassIVE.quant provides more category for quantification and metadata. The metadata for description of the sample and the experiment design, quantification for quantification result, and then statistical analysis result for statistical analysis for quantified analyte. So now there's a way to add the metadata and quantification to the data set. In addition, any other intermediate output to reproduce the research, for example, the parameter file or R script can be submitted too. Second, the MassIVE.quant provides the branch structure for reanalysis in a data set. As we talk shortly, different combination of tools and the method that generate the different intermediate file and conclusion. The branch structure allow us to submit the unique conclusion with the all intermediate output as an analysis under the data set. There are 3 layers of repository structure. Dataset is set up for each experiment or publication. It has a unique MSB ID. Container can be set up under each dataset to aggregate groups of related reanalysis. Each container has the unique container ID. Reanalysis is set up for individual reanalysis and also has a unique reanalysis ID. The dataset can have many containers, and each container can have many reanalysis. This layer allows us to organize reanalysis in different or even efficient ways. Here is the example of reanalysis branch in one dataset. The dataset called MSB79843 has one control mixture experiment including 12 raw files with the 4 conditions. Originally, the raw files were submitted in this dataset. Then we reanalyzed these raw files with 10 combination of identification and quantification and the different parameter. And then these 10 reanalyses were grouped in the first container. Next, we reanalyze this quantification with the different statistical approach. There are 12 new reanalyses and grouped in the second containers. Here is another example. So this data set was originally submitted PRIDE. The data set submitted in PRIDE can be imported to the MassIVE, and now it has MassIVE ID. This data set is for one publication, which includes 3 different experiments. Previously, it is not easy to separate all files for each experiment. So we set up the container for each experiment. Now we have a 3 container for each experiment. Then for each experiment, we submit the reanalysis, including all the intermediate file up to the statistical analysis. Moreover, the MassIVE.quant provides a summary for metadata and quantifications and statistical analysis result in different repository layers. In dataset, there is a summary of the dataset level, such as original submission and then the reanalysis and how many reanalyses are there. For example, here for original submission by owner, the no metadata and identification result and quantification result are submitted. However, for next of the 5 reanalysis include all this information. In container level also there's a summary. For example, this case there are 2 reanalyses are involved in the container, this container, and then it shows the summary of the all information. In reanalysis level, summarization also there. So for example, for this specific reanalysis, there are 3 conditions, there are 12 raw files, there are 3 conditions and 12 biological replicate, and then this much number of the quantified protein, and among them this much number of the proteins are differentially abundant. And all files are available to download through the FTP. In each reanalysis, the MassIVE.quant support the interactive result view for quantification and then metadata. The MassIVE.quant currently provides the compilation of the 46 public dataset. It includes 10 control mixtures and 36 biological studies by different acquisition types and the experiment design, and 187 reanalysis by different processing tools, algorithm, or options. To add the dataset or reanalysis to MassIVE.quant, the files should be submitted in the 4 collections: raw files, which means raw files submitted in the raw file collection, and the metadata in metadata collection, and the quantification and statistical analysis of the quantified analytes. In addition, we recommend to submit additional information such as parameter setting for identification and the quantification analysis, and script for statistical analysis in method and protocol collections. In order to scale the submission procedure and to ensure the reproducibility of the quantitative workflow, MassIVE.quant maintained the data set with the 4 levels of the curation: bronze, silver, gold, and platinum. During the submission, the infrastructure checked whether the submission of the dataset or reanalysis meet the minimum requirement for the entry level of the curation. The minimum requirements are the files in the 3 collections: metadata, quantification, and statistical analysis. Especially the metadata should include the raw file information matched to partial or whole file name in the raw file collection. Also, it should have the condition or biological replicate for corresponding raw file at the minimum. To level up, the submitter can request advanced review to MassIVE.quant team. The next level is a server. The MassIVE.quant team manually review whether the information is correct. For example, biological replicate is correctly or annotated or not, or there is any missing information, for example, technical replicate or fraction information if they are needed. Files in the quantification or statistical analysis can be parsed and show up the biasing or not, and so on. Gold and platinum, the levels are based on repeatability or reproducibility. The gold level tells that there are enough information provided in order to repeat the same data analysis, such as the parameter information, including the version of the software, algorithm, configuration file, script, or tool-specific file as alternative. Platinum level means that anyone can reproduce the same conclusion for publication. For example, full documentation for whole data analysis from the raw file to the statistical conclusion for publication will be in this case. When researchers submit all the required metadata quantification statistical analysis, the MassIVE.quant summarized information about experiment design and the result too. So for example, here for this specific experiment, based on the metadata, they just count condition and then replicate and technical replicate information. And from the quantification and statistical analysis, they also summarize this information. So now there are 46 public data set in MassIVE.quant. Among them, 25 data set with the platinum level, and then 18 data set with the gold level of the curation, which go all the way from raw file to statistical analysis. Also, this collection includes many reanalysis by different processing tools or options for each experiment. The different processing tools output different types of file, and it is not easy for researchers to submit. So this collection, especially for the control mixture data, can be as an example to show which output of the data processing tools or which intermediate files are important for reproduce the result and need to submit the MassIVE.quant. Next, the MassIVE.quant can automate the statistical analysis or compare the result. If required, the metadata and quantification are provided in the MassIVE.quant. MS-Stack workflow is example of executable workflow for MassIVE.quant, which can run MS-Stack, which is our package for differential abundance analysis, using MassIVE.quant interface. There are two ways to use the MS-Stack workflow. First, user download all the raw file and FASTA file from the data set to your local computer, and then use any software to searching the peptide and then protein identification and then quantification. Then upload the quantification and metadata to the MassIVE.quant, and then use the MS-Stack workflow for differential abundance analysis. And then automatically you can submit the output of differential abundance analysis as a new reanalysis. Or user can reanalyze the submitted quantification in the data set and then submit the result as a reanalysis under the same data set. Also, the comparison workflow can compare the statistical analysis result across the different reanalysis. For example, here is an example we talked about before. Under the first container, there are several reanalysis quantify the different data processing tools. The comparison workflow compare across up to 5 statistical analysis results. To compare statistical analysis result should be submit the MassIVE.quant, and then this workflow automatically can read the statistical analysis, the result, and then compare. The last, the documentation and video tutorial for each workflow and step and different cases are provided. All links are available in the MassIVE.quant webpage. In conclusion, the MassIVE.quant is an important and necessary step toward reproducible quantitative and mass spec-based proteomic research. When it is possible to automate the workflow fully, the repository provides the infrastructure for doing so. The collection of the consistently documented and then analyzed benchmark dataset will aid in future development of the data analysis method. I'd like to thank all collaborators in our group, especially the MassIVE team from the Dr. Nuno Vandela's group in UCSD, who made this MassIVE.quant possible. And thank you.